Hello, everyone. Good afternoon, and welcome to the Northeast Aquatic Plant Management Society's Autumn Webinar Series. This is session 2 of the series, which is our Spotlight on Species. Today, we are looking at grass carp. So a little bit about the Society. The Northeast Aquatic Plant Management Society's mission is to assist in the management of aquatic vegetation, to provide for the scientific and educational advancement of members, encourage scientific research in all facets of aquatic plant management, and to promote an exchange of information among members and to extend and develop understanding in this discipline. Our members are from academia, government agencies, private consulting firms, herbicide manufacturers, and lake associations. And we have annual meetings, annual plant camps, webinars with CEUs like the one you're attending today. And we also offer student scholarships up to $10,000. And you can visit www.neapms.org to learn more about us. My name is Lindsay Yoder. I am the moderator for this session. I am the Aquatic Invasive Species Coordinator for Region 9 of the New York State Department of Environmental Conservation. And a little bit more about this webinar. As I mentioned, this is the Spotlight on Species, Session 2 on grass carp. This webinar is set to run from 1 p.m. to 3 p.m. There will be 2 presentations with a block of about 15 minutes at the end for questions and a discussion with the presenters. And so I ask that you type your questions into the chat box as the presentations are going, and then at the end I will read them during the Q&A session. And please note that we are offering CEUs for this webinar, so make sure that you check in with Summer Weidman and answer the quiz questions. And this webinar will be recorded, and it will be shared via link on the NEAPMS website with more information to follow. So we have a jam-packed webinar for you today with some awesome talks. Up first, we have Patrick Kachowski and the USGS team talking about their research in support of grass carp management in the Great Lakes and the upper Mississippi River basins. And following that, we will have Ryan Young with the U.S. Fish and Wildlife Service discussing ongoing response efforts for grass carp in the Laurentian Great Lakes. So I'm going to stop sharing my screen. And Patrick, you're good to start.
Great, thank you, Lindsay. Thank you for the invitation to present at this autumn webinar. Again, my name is Patrick Kachowski. I'm the Aquatic Invasive Species Program Manager for the U.S. Geological Survey. USGS has been conducting grass carp research in the Great Lakes and upper Mississippi River Basin for about 10 years. This is funded by Congress and through the Great Lakes Restoration Initiative, so from Congress directly by appropriations to the U.S. Geological Survey, and then through a competitive process to secure additional funds through the Great Lakes Restoration Initiative. From the beginning of the program to conception, we started with not even knowing that grass carp were an issue, to having developed multiple ways of estimating where they spawn and estimating what tributaries are using and determining where they're spawning to assist in removal efforts. And there's a lot of modeling behind that, and our researchers today will step you through the work that they've done to help inform management efforts for grass carp. So that's all I'm going to say. I'm going to leave as much time as I can for our scientists. And again, thank you for the opportunity to present. And I'll hand it over to our first speaker. Thank you.
Okay. Can folks hear me?
Yep, you're good.
Okay, thanks. Just want to make sure. So I'm Corbin Hilling. I'm a research fish biologist at the USGS Great Lakes Science Center Lake Erie Biological Station. Get some more light. There we go. And I'm going to be talking about grass carp early life history and leading into presentations from Steve Spear and Ryan Jackson, who are also with USGS, just different centers. So I'm going to jump right into this. So I'm focused on early life history of grass carp in the Great Lakes specifically, and there are a ton of partners and a lot of different moving parts to this. So grass carp in the Great Lakes are managed by the Lake Erie Committee, which is made up of the representative fishery management agencies. And that management is very collaborative, and part of that is facilitation through the Great Lakes Fishery Commission. And they're provided information by the Grass Carp Advisory Committee, and there was a structured decision-making exercise several years ago where folks all got together and kind of hashed out what they were looking for in terms of grass carp management and how to manage grass carp and how that would really all work. So getting down a little more into the nitty gritty, we've kind of, Rob Hunter, a PhD student at University of Toledo who now works for USGS in our office, put this schematic together of a three-prong approach to informing and managing grass carp, where we have removal efforts, there's early detection work going on, and there's also research, which has generally been grouped into four categories here, but this isn't all inclusive. Early life history, telemetry, population modeling, and more novel technologies, which we're going to hear about momentarily. And again, you can see here a lot of partners working together collaboratively. So in terms of grass carp early life history, grass carp are pelagic spawners. They look for turbulent areas in rivers. And so they rely on currents to move their semi-buoyant eggs downstream to habitats that are suitable for rearing. And so they require stream flow, or it's possible that eggs could settle on the stream bottom and perish. And that provides an excellent opportunity for using early life stages as an early detection tool, where we can use these bongo nets, as you see here, to filter eggs and larvae out of the water column, and then collect biological data from those, which is useful for other efforts. So this is a plot of BongoNet effort through time. This program started with Patrick initiating things in 2014 with some pilot surveys in the Sandusky River. And through time, we've expanded the systems that we're covering. We now cover six rivers in Ohio and one stream, one of those being a stream in Michigan that flows into Lake Michigan, the St. Joseph River. And you can see effort has generally increased through time. We're covering more systems, and here's a quick snapshot of what we've found through time. So surveys started in 2014. The first grass carp eggs were collected in the Sandusky River in 2015. 2016, we didn't collect any grass carp eggs at least. It's a low water year. They often spawn in response to high flow events, which further helps get water velocity and turbulence up enough that those eggs can stay off the stream bottom and move downstream to those preferred habitats. The first eggs were collected in the Maumee River in 2017. The first larvae were collected in 2018, which was a major indication there. Given that the Great Lakes tributaries are not these like extensive riverine networks like we see in the Mississippi Basin, it was never necessarily clear that eggs were going to make it in suspension long enough and not go into a lake ecosystem and settle to the bottom. So collecting larvae in the Maumee River indicated that hatching in the rivers was indeed possible. Nothing in 2020, limited sampling effort for reasons you all can probably guess. Moving to 2022, the first larvae were collected from the Sandusky River, making 2 rivers that can support in-river hatching, and we collected eggs in the Huron River for the first time. We documented spawning later than we maybe would have anticipated in 2023 with a late flow event in August in the Sandusky River. And this year, again, we've expanded our known spawning period with collection of eggs on May 1st, which is now the earliest we've observed grass carp eggs. So in terms of early life history being a contributor to early detection, we documented spawning in the Huron River when a single diploid adult was known. So we have collected more eggs than adults in the Huron River at this point, just highlighting how important this is. The eggs can't move. The adults are kind of elusive, which Ryan, I'm sure, is going to talk about in a bit. They have to go downstream, and this provides a useful way to understand where populations are, where fish are spawning. And Ryan's going to talk about some useful uses for the data that we're collecting. Some other insights. We've defined what spawning conditions look like with high flow events and temperature thresholds. Again, noting that spawning season is May to August. Provides removal crews a good opportunity to target fish when they're vulnerable to electrofishing. They're moving up into shallow areas to spawn. We can predict when those are, and we now know that that's somewhere between May and August. We're working to understand gonad maturation timing, so when are fish ready to spawn, and what factors influence that. Some otolith microchemistry work has indicated that most adults are produced from the Sandusky and Maumee rivers, with the possibility from other systems, and we're doing some work to understand where recruitment's possible. And then the big thing from this is, even though we're looking at spawning, these data that we're collecting are useful for other purposes, and Ryan and Steve are going to talk about some of that right now, so I'll pass it off to them.
All right, thanks, Corbin. Yeah, I'll just have you, yeah, thanks. So yeah, I'm going to talk about work that we've been doing in using environmental DNA to help identify spawning activities, or in evaluating if it can. And I don't have time to go into a lot of in-depth about eDNA if you're not familiar with it, but in general, what we're talking about here is filtering water, and then cells and other genetic material is caught on those filters. And then we can process that, at this point, process it in a lab and use targeted genetic markers that in this case will only work for grass carp. So we're able to see if grass carp DNA is present in those filters. And eDNA has been used for a lot of invasive species surveillance, early detection, and other objectives. But for looking at events such as spawning, especially as Corbin talked about, you'll hear more about with the spawning with something like grass carp that can be unpredictable. It's related to certain conditions like discharge in the river, that it's hard with manual sampling or going out occasionally to detect those sorts of things. So automated regular eDNA sampling could really be key for helping to basically be an alarm for events like spawning. And so you need samplers that can be programmable and that on their own will filter water and store those for environmental DNA analysis. So store those in a buffer so that the DNA wouldn't degrade over time. And so we've done a couple of sessions in 2022 and earlier this year in 24, working with the Monterey Bay Aquarium Research Institute, who's developed a sampler. And you can see that large 50-gallon drum in there, that is a sampler with a bunch of filters. And then we have a water intake. And so it will store there. And it can store up to 132 filters. And we can set a custom program for how frequently. And we can actually change that on the fly. It has connection to the cell network. So if there's like a high flow event, we can say, okay, let's take 3 filters today, instead, even if we had previously programmed it for like 1 or 2. At this stage, with this type of sampler, we aren't accessing or getting the results in real time. We're taking them back to the lab and then analyzing them and looking at basically back in time if it was concordant with known spawning events or discharge events. But I'll talk at the end about how that could work being done to make that more in real time. So next slide. So I'm going to talk about samplers on the Sandusky, as I mentioned, which is, as Corbin talked about, is one of the confirmed spawning tribs to Lake Erie. This map just, it's northern Ohio, and this map just is to show you that we've had the sampler in 2022 and 2024 in 2 different sites, and also kind of the direction of downstream movement toward Lake Erie. And 2022, the sampler was several kilometers, 15 to 20 kilometers downstream of the known spawning site. That was really driven by logistics of placing that sort of sampler. We had to have some sort of infrastructure. However, in 2024, the infrastructure was there and we were able to place the sampler essentially immediately downstream of the known spawning site. In 2022, we deployed it from early April to mid-June, sampling 1 to 3 times a day, depending on conditions and time during the season, with the most intense sampling happening in late May to the first half of June, when previous work had suggested was the kind of the hot spot for likely spawning and fish aggregations. In 2024, we, in addition to the difference in site, we pushed back the deployment to starting in May 8th to allow us to go further into the season, which at the time we thought would increase our chances of detecting spawning, so into early July. But we kept the most intense sampling at about the same time. So next slide. So these are the results that we've gotten, and so these graphs for the next 2 slides. On the y-axis on the left is eDNA detection. So it's just whether eDNA was detected or not, a 1 or a 0. And so those blue circles, if they're at the top, that means at that point in time, at that date, which date is on the x-axis, eDNA was detected for grass carp. If they're on the bottom, it means it was a negative. The orange dots represent the discharge. So you can see across the sampling period peaks of higher discharge that would be expected to be conducive for grass carp spawning, and when it's not. And then you can see the X's represent times when the egg sampling crews from University of Toledo and others were going out looking for eggs, and X means they didn't find any eggs. Circles were the 2 occasions where they did find eggs in 2022. So what was exciting about this is you can see, well, first off, oops, sorry, if you can go back, sorry. Only about 22% of our samples were positive, even though we know that fish are present in the river year round. So this is telling us that we're not picking up every grass carp that's moving through. From an early detection standpoint, that wouldn't be great, but for something where we're trying to detect an aggregation sort of activity, that actually is probably a bonus. And the reason I think we're not getting as much detection, even though we know fish are there, is that turbidity and not being able to filter as much water. But I can talk more about that if there are questions. But we see even with this, there's clear associations with discharge. You can see the cluster of positive points when those hydrographs go high. And especially exciting, the two spots where there were eggs detected, you can see a run of multiple positive samples in a row. Okay, next slide. In 24, there were some similar patterns and a little bit different. Similar was, again, we only had 20-some% samples positive, even though, again, fish are present there year round. We saw very clear, so there were less high discharge events this year, but we see a very clear association with that. The 2 peaks of discharge are immediately followed by several runs of consecutive positive samples. So an even clearer pattern, I think, than last year, even though there were fewer events. However, egg detection, we don't have that matched up like we do in 2022. One we missed, as Corbin mentioned, there was a May 1st spawning this year, and our samplers were out a week later. So that was unfortunate. But also when I don't have the egg sampling events on this graph, but when the egg sampling was happening, wasn't quite concordant with the positives. For instance, there was an egg sampling event that did not detect any eggs on May 11th, but that was just before the pulse and when we got a bunch of positives. So there weren't egg sampling out during the times where we had those. So it's hard to say if there was actually spawning. Other than that, it fits with the discharge patterns. Next slide. So moving forward, I think what's really promising from what we have thus far is I think there's really strong evidence for having these consecutive positives represent almost certainly aggregations, and in some cases at least demonstrated spawning and perhaps spawning events that were missed by other methods, which shows that I think this can be, that sort of pattern could really be a key to focusing on doing sampling during then or capture events. Obviously, for that to be useful, we can't wait to bring them to the lab. They need to be known in real time. There's currently more portable and flexible autosamplers that are available or will be available very soon. This one was very large, the 50-gallon drum, and hard to install, but that's changing, and there's some already on the market. Currently, it could be combined with some rapid eDNA testing that our lab and others have been developed, including for carp. In the future, and hopefully the near future, there would be automated eDNA analysis where all of this happens in the sampler completely with no manual input, and then that could send a signal through a cell network. And as soon as you had 2 or 3 hits in a row, that could trigger an alarm or warning to folks. So it's obviously still very much in the research phase, but the results thus far are pretty exciting for doing this sort of monitoring. And I'll pass it on to Ryan next.
Thanks, Steve. So one of the things that we can do with the eggs and larvae that are collected is understand more about the processes of reproduction for grass carp using drift modeling. And essentially, drift modeling combines the biology and physics, so the life history, development, and physical characteristics of eggs and larvae, along with the hydrodynamics of rivers, infection, turbulence, dispersion. And with drift modeling, we can give management agencies a number of well, quite a bit of information, you know, identify suitable spawning rivers, understand conditions for potential recruitment, identify or inform when crews should go out and sample, and identify grass carp spawning areas, which is one of the focuses of the next couple of slides. Next slide, please. So we do this using the fluvial egg and drift model. So this is called FluEgg. It's a custom model that we built that basically includes all the processes, the biological and physical processes that are important for transport and understanding that drift period all the way from spawning through, you know, egg growth and changes in property over time, through egg hatching, which is an important phase, and then gas bladder inflation, which is a stage at which the larvae then leave the drift in search of nursery habitat. Next slide, please. So using FluEgg, one of the advantages of this tool is that we can actually, through the captures of eggs and larvae downstream, through those samples in the bogland nets that Carvin was talking about, we can use those together with developmental data and developmental staging of those eggs and larvae by the biologists to then backtrack and use our FluEgg model to identify where the potential spawning occurred. And this is all because of the fact that we can glean information, knowing water temperature and developmental stage of the eggs. We can actually back calculate the time at which the eggs were fertilized and then use the physics to backtrack where those eggs and larvae were spawned. Next slide, please. So there's a number of ways to do this. It's been approached from using two different methods. Essentially, first method uses forward runs of FluEgg. It's an iterative method that basically chooses spawning locations upstream and then looks at where that plume is relative to the egg capture location and iterates and kind of steps downstream until you have the best, highest probability, I guess, for eggs being at your capture location at the capture time. And that gives you a potential spawning area. The second method uses reverse particle tracking, which is basically, it's built into FluEgg now, and it essentially runs the model backwards from the capture location, the capture time, all the way back to the fertilization time. We actually switch the flow in the model to run upstream. We reverse the growth of the eggs and reverse all the processes. The one thing we can't do is reverse dispersion. So we still end up with a distribution of potential spawning areas. But it requires only a single simulation, whereas the previous method requires a whole suite of simulations. Next slide, please. So we've applied this to the three tributaries of known spawning in Lake Erie, Sandusky, the Maumee, and the Ohio River. It's been successfully predicted two of the spawning areas within the Sandusky River. There's a primary and a secondary. Those have been validated and confirmed through additional modeling as well as field experiments or field observations. And with the removal of Belleville Dam back in 2018, it opened up a new section of river in the Sandusky. We've recently reevaluated the spawning locations and identified potential new grass carp spawning locations upstream of where the dam used to be. In the Maumee River, we've identified over 12 potential grass carp spawning areas over a 77-kilometer reach of the river using 3 years of ichthyoplankton data from Corbin's group and others. And then, of course, with the Huron River, we've been able to identify a potential spawning location for those 2022 eggs that Corbin talked about. In this case, we use the National Water Model from NOAA to help drive FluEgg. Next slide, please. This comes full circle because we can take all this information from our modeling and the egg and larval captures and then put it back into a predictive model, a forecast model, to help forecast when grass carp spawning is likely to occur in these systems based on observations and all the data that we have. And so we have USGS SpawnCast, which then can help predict up to 5 days out when spawning events are going to occur. So we can management agencies can put their crews in the right places at the right time for either sample collection of eggs and larvae or collection of adults to try to knock down that species and harvest the adults before they have an opportunity to spawn. I think that's the last slide. We can hand it off. Yep. Thank you. I think Matt's up next.
Can everybody see my screen?
Yep, and we can hear you too.
I'm going to start this presentation off. My name is James Roberts. I'm a research fish biologist with USGS at the Lake Erie Biological Station. Matt's going to bring the second part of this presentation, and he's also a research fish biologist. He is at the USGS Columbia Environmental Research Center. I'm going to turn my webcam off because I was having some issues with watching the previous one for the remainder of this talk. But just to let you know, this is what it looks like. So we'll talk today about grass carp telemetry and how we use that to inform potential deterrents and also removal efforts for this invasive species. Next slide. And the approach we're using is trying to inform invasive species removal by understanding aggregations of these individuals. This is also referred to as kind of like a trader approach. We use tagged individuals to better understand their movement, behavior, and habitat use. This hopefully can lead removal crews to grass carp aggregations. This has been used in invasive species management, probably most notably and kind of first reported with goats in the Galapagos Islands. We try to do this both in real time, where there are real-time receivers that provide alerts when tagged grass carp are in certain areas, and then also satellite tags, which provide locations when those tags are able to get satellite connections. So we'll give some examples of that later today. And then we also do it using predictive analyses, which try to describe spatiotemporal patterns in habitat use of grass carp, and then point removal crews in to those areas given the time of year and other conditions. And we also try to understand the behavior of grass carp when they're in these specific habitats to disrupt critical life history events. An example of that we'll talk about today is to deter spawning migrations and behavior, which was kind of identified during the initial structured decision-making for grass carp management in Lake Erie, trying to kind of deter and disrupt spawning in the Sandusky River was a kind of a key management goal. Next slide. And to do this, I've mentioned telemetry, but specifically we're using acoustic telemetry. And this is a technique where a tag is implanted. You can see the tag in the middle of the slide here in the body cavity of these fish. And these specific kind of tags give off an acoustic signal, which is encoded with a unique ID. We then put receivers throughout the system. We're interested in detecting these fish that listen for these acoustic signals, record the unique ID, and then also give it a date and time stamp. So far, this has resulted in 121 successfully tagged grass carp. Currently, there are 60 active tagged grass carp within the Lake Erie Basin, and this whole process started similar to the early life history sampling like Corbin was talking about in 2014. We're using a V16 model from the Nova Sea. This tag has 120-second nominal delay. And so every about minute to 2 minutes, a acoustic signal is given off from this tag, and these tags last for up to over 6.5 years. And so far, we have over 8 million detections, and the amount of information we get from these tagged fish is just increasing with time. Next slide. So to listen for these tag fish, we have a tiered array of receivers within Lake Erie, and they are shown here on this map. The basin-wide array, which really covers the nearshore habitats of Lake Erie, is shown in red. We have a tributary array, which covers the rivers and specifically focuses on the known spawning rivers. That's shown in blue here. And then we have our alert array, which is shown in the different types of yellow points that gives us alerts in more of a real-time fashion as these grass carp move into specific habitats. The other types of receivers give us the information on an annual basis. So we have to visit these receivers and download the data, and then we're able to put that all together to analyze what happened the past year. And in addition to those receivers I just mentioned here, this is also embedded in this larger GLaDOS array, which stands for Great Lakes Acoustic Telemetry Observation System. And so there are some other projects and other kind of base receivers that are located throughout the Great Lakes, and the white dots here are showing that GLaDOS array in Lake Erie. So you can see that in addition to our expansive grass carp specific array, we also have ears, we also have receivers in other parts of Lake Erie to inform us of grass carp protections. And also as part of that tributary array, we have these fine scale arrays, and that's shown here on the bottom right of this slide. And this is specifically in the Sandusky River, so one of the known spawning rivers that Ryan was talking about here. And this is the first one where eggs were detected. The river kind of flows from the bottom of the screen to the top. And these fine scale arrays have receivers that are very close together. And so normally in the other arrays, when you get a detection, you know that the tag was within half a kilometer to a kilometer of that location. And you just provide the location of that receiver as the location of that fish. When we have these fine scale arrays, the receivers are close enough that one signal from the tag can be detected on multiple receivers, and then we can kind of triangulate where the fish actually was. So get some synthetic 2D locations of these fish, not just the location of the receiver that the fish was detected on. So that gives us some more insight into the behaviors and the movement patterns of these fish. And this is important in a habitat like this, the Sandusky River, where critical life history event spawning takes place. Next slide, please. So some kind of broad takeaways from the telemetry work that's been going on so far with grass carp. Most of the detections of the grass carp are in the Western Basin, and the figure here on the right is showing all the gray dots are detections of grass carp, and the white lines are connecting the individual kind of detections of these fish. There's one grass carp that moved from Lake Erie into Lake Huron. Grass carp are not limited to just the spawning tributaries. They've also been detected in other tributaries of Lake Erie. The mean total movement distance across all these individuals in a publication from 2001 is 263 kilometers, so they can move quite large distances. There are also seasonal patterns in these detections where we see more movement in the warmer summer and early fall periods than in the cooler winter periods. And there's also been detections in all three basins of Lake Erie. Next slide, please. So next, I'm going to show you an animation here that kind of combines the telemetry and the early life history information that we heard of earlier. So we talked about when grass carp eggs were detected, and Ryan showed how that can be used to look at where the eggs were first deposited in the river. We also took those egg collections and we looked at what the behavior was of these grass carp within the Sandusky River when that happened. And so this was an egg collection from August 20th of 2023. On the figures on the right, we're going to show some abiotic conditions over this time period: discharge of the stream on the top, temperature of the stream, and then atmospheric pressure on the bottom. And that gray bar in the middle there is the day, August 25th when eggs were collected. And then on the left here, this panel, again, this is the Sandusky River. The top right of this map is Sandusky Bay. And so it flows from kind of the bottom of this to the top. And then there'll be uniquely colored dots that move along. And those are individual grass carp over this period. And so this is going to show you 5 days prior and 5 days after this spawning event occurred. So hit the movie, Matt. And you can see a lot of the detections start in the lower part of the river. But as the flow starts going up and it gets closer to that egg collection, we see a lot of the fish moving up to this upstream area. This is the Brady's Island area. And then we see them kind of moving back down into that lower river after the spawning occurs. And so this is important, and now shown in red here, this is a proposed barrier location, which is just downstream of the primary spawning location that's been determined by the flu egg modeling, which is around that Brady's Island area, just upstream of that red thing. And so this information was useful in determining, where potential barrier or deterrent could go, exactly what that deterrent might look like, and if it is going to be installed is still under development. But information like this will help and has helped, determine when such type of a deterrent would be helpful, and that this would be a good location to prevent grass carp from accessing that primary spawning location. But beyond that, we also saw some interesting patterns from this animation, where a lot of these fish were found more frequently in that lower part of the river. And so this might give us some insight into where removal crews could be focusing their efforts when a spawning event isn't occurring. And Matt's going to tell us some more about using data like this to look at, where we might be good at catching fish, where we're not, and some other kind of behaviors that may be able to be focused on by these removal crews to capture more grass carp. So I'll hand it off to Matt.
Thanks, James. So we're going to dig into some of the two-dimensional data, but also look at general patterns and focus really heavily on this one known spawning river. So we're going to combine capture data, which Ryan Young later in this seminar is going to go into great detail. So I'm going to skip over some details on the capture part of things, but these are the removal events that happen to get rid of grass carp in the Lake Erie Basin. And then within the Sandusky River, we have this year-round array. These are those kind of passive gate receivers that James had mentioned. And then we also have these two-dimensional VEMCO positioning system arrays as well that give us that two-dimensional data. And so we're going to try to combine these data. But before we do that, we need to wrap our heads around this idea of catchability, which is really a pretty simple concept. It's a catchability coefficient, or Q, is the proportion of a population that you would expect to capture in a single unit of fishing effort, right? So if you take this completely realistic pirate ship electrofishing example, going out into some area to catch fish, if there are 10 grass carp present and it catches 3, then your catchability coefficient is 0.3. So you got a Q of 3, right? And the reason that I bring this up is because I want to mention a very common formula that we use in fisheries ecology, and that is the catch per unit effort, or C divided by effort. And oftentimes we set this equal to abundance. But in reality, this is actually a relative abundance, and even more so in reality, we often equate out the catchability coefficient by just setting it equal to 1, so we don't have to deal with it. But we know that the catchability coefficient changes over space and time, and also with the different gear types. So in the grass carp program, there are many different gear types that are used, a combination event where the nets and electrofishing are deployed, just electrofishing, as well as hoop nets, and some others that Ryan Young will talk about later. And we know that there are different catchability values for each of those types of gear, right? So what we can do is we can rearrange this equation that we had up there above to calculate the catchability for each of these gear types. The problem is that we don't have a known population size, right? So we don't know what to set n equal to. But what we can do is we can leverage the telemetry data. So we know exactly how many fish are in the river at any given moment, right? So we have a known population size, and we've captured and recaptured some of those fish occasionally in each of the gear types we used. The problem is we can't use the exact capture numbers anymore because we can only use that subset of telemetered fish. But that's okay, because once we calculate the catchability coefficient, we can reapply it back to the total captures. So what in reality catchability looks like for grass carp, at least in the Sandusky River, across these three gear types, is really, it's pretty close to zero. It's very, very low. So they're an elusive fish. So, taking this pirate ship example again, if that same ship was to go out and electrofish for grass carp, and there are 10 fish present, most likely across all of these gear types, you're going to catch 0 of those fish on most occasions. But if you do it enough times, you will eventually catch them. So what we can do now, though, is take those catchability coefficients and put them right back into the original equation, right? We can just reformulate this again and calculate for n, right? And now we can use all of the capture data that comes from the removal cruise and start to create these abundance estimates to evaluate the management program within at least the Sandusky River. And by and large, all three of these sampling techniques tell us a similar story, and that is that there is this reduction in abundance within the Sandusky River. But now we want to start to understand how we can help to improve that. How do we become more efficient at capturing fish? Well, what you're looking at here are polar plots. They tell us how many fish are in the river using the telemetry data by month and hour, mean hour of that month. The takeaways here is that as that circle gets bigger, so like these blue, these blue dots and circles over here, for example, that's one of the circles. Here's another one. As those get bigger, it's a representation of more fish in the river, and that's represented by this axis over here at 3, 6, and 9. The take home here is that there are more fish and it's more stable in the winter months. So November through February, so there are more fish within the river. And the other take home here is that as that point changes color to red, that's an indication that there are more fish comparatively that month by that hour. And so, you think about these kind of like diel shifts that might be happening within the river by month. Well, what we start to see is that there are more fish coming into the river, into the main stem of the river during those summer months, as indicated by those red dots. And that's generally happening between the hours of like 11 p.m. and 8 a.m. So overnight into the early morning hours, right? Well, that's unfortunate because most of the sampling doesn't happen at that time. But these are just general patterns, right? Can we actually model this? So what we've done is taken all of the capture data, all that telemetry information now with behavioral metrics, we put it into this generalized additive model to see if we can't describe some of the partial effects on capture. And that's what you're looking at here are the results. The most important variables in this model ended up being hour of the day, day of the year, the density of telemetered fish count, or the abundance rather, and then the number of sampling events. For hour, what I want you to notice is that anything that's above these blue lines represents a zero catch, an actual capture. Anything below is a zero capture event. The majority of zero data comes from midday. This is when we're least likely to catch fish. And you can see those tails start to go up in the early morning hours and late at night. There are a couple of other peaks in capture that happen in late May, so late spring, August, and early November. And then if you assume that telemetered fish represent the population, the whole population of grass carp, you would kind of understand that this threshold of 5 fish entering the river and more results in more capture, right? Because the abundance of fish is presumably getting bigger within the river. And then there's this also threshold of sampling event, essentially telling us the more effort we put into it, the more likely we are to catch fish. And that's probably pretty intuitive. But so we know that capture decreases in the middle of the day, but this has us kind of questioning, where are they going? Why does capture fall out like that middle of the day? So the details of this model and some of the other stuff I just presented, you can find in this paper we put out just a little bit earlier this year. But the follow-up to this is, where are they going, right? So postdoc on the team here, Dr. Sophia Bonjour, is running these hidden Markov models, which allow us to understand these kind of latent behaviors or hidden behaviors. And the way to kind of think about this is, sometimes fish are moving really far and fast. Sometimes they're hanging tight. And then, they might go back into that far and fast kind of behavioral pattern. Well, we can separate these two statistically using these hidden Markov models to try to make inference about where they're doing this and when they're doing this. So what we want to do is think about like the spatial distribution of backwater habitats and when they're switching from that kind of longer state to that shorter state, right? Because that longer state is going to indicate that they're moving really far and fast, shorter state kind of hanging tight. So we've classified all of the points within the Sandusky River either near backwater habitats or in them. So that's like the yellow and the orange, or within the main stem of the river out of backwater habitat. So that's all of the red dots. These backwater habitats, at least within the Sandusky River, are made up of kind of this Phragmites, real shallow habitat, lots of North American lotus, so submergent, emergent vegetation. So it's generally a place that they may go to seek refugia from the high water velocity within the main stem. The results from these hidden Markov models are really interesting because what they're telling us is that they're more likely to be in state 1, so that shorter step length, that yellow line, whenever they're in and near backwater habitats. So that's these top 2 figures, in backwater habitat, near backwater habitat. When they're out of backwater habitat, they're way more likely to be in that state 2. So they're moving farther and faster whenever they're out of the backwater habitat. The other take home here is that this is a nearly inverse relationship with capture, right? So if you think about the probability of being in state 1, that short step length increases middle of the day. So you got hour on the x-axis. Well, that's an inverse of our capture probabilities as well, right? So essentially what we think is happening here is that they're unavailable for capture whenever they're moving into these backwater habitats. The strike crews can't really go back there and catch them very efficiently. So we think that, we had pitched this to the removal crews that we need to take advantage of some of these points of constriction, either when they're coming or going from backwater habitats, potentially targeting certain temperature profiles. And we also thought it was a really good time to start exploring alternative capture methods. So either blocking backwater habitats or setting passive gear overnight when the fish are far more active, more likely to be in that second state, moving far and fast. And with those recommendations and some innovative thinking from folks at Fish and Wildlife and elsewhere, we've been able to increase capture this year as a result of some of this modeling work 2.5 times. So you'll see some of that from Ryan Young a little bit later. But there's one limitation. Well, there's a couple, but the major limitation of acoustic telemetry is that it requires a network like James was talking about, the Great Lakes Acoustic Telemetry Observation System, or a really fine scale positioning array, which is relatively expensive. And it also isn't possible to have that kind of coverage everywhere. So what we've started to do is explore these satellite tags, and that's what you're looking at here is a screenshot of a few different models of satellite tag that we put as a harness on top of these fish. And whenever it breaks the surface, it sends a signal out to satellites and we get a position. And this has been really useful for removal crews to inform their daily activities. And it's also been pretty interesting to see how some of these fish over the last year have been using space where we were unaware. So an example is this fish in yellow, which is the only one that we've had tagged for longer than a year now. What it told us was that it was spending a substantial amount of time outside of an array or outside of any receiver coverage using harbor habitat that we did not know that they were using, sampling the lower part of the river and then going back out. So essentially unavailable for capture the majority of its time over there in the Sandusky River. And so this is allowing us to kind of like think about innovative and exploration into different types of gear in different places. I'll stop there, and if there are questions later, feel free to reach out to us on email or at the end of this talk. Thanks.
All right, and the next team, we are running a couple minutes behind, so if y'all could just keep it to a tight 15, but we can have the next team go.
All right, assuming everyone can see that and hear me, I will be giving a brief overview today of our work on a couple deterrent systems. So you heard some of this mentioned with the Sandusky River. What we're looking at are deterrents for upstream movement of fish. I should introduce myself. My name is Mary Beth Bray, research fish biologist in La Crosse, Wisconsin. And Andrea Fritz, who's also on, leads the work on the bioacoustic fish fence project. All right, so what we want to do, what the goal is to reduce upstream movement or movement of invasive species, specifically passage, often at locks and dams. We want to be able to facilitate passage of native species like paddlefish and sturgeon. And what we know about carp is that they're a stereophysan fish, so they have this enhanced hearing. They have what we call a Weberian apparatus, connects their inner ear to their swim bladder, and this makes them more sensitive than a lot of other fish to sound, to vibrations, things like that. And so we've used that to develop an underwater acoustic deterrent system, and this is a series of 16 underwater speakers that we installed at Loch 19 in the upper Mississippi River, right at the southern corner of Iowa, bordering, sharing the lock approach channel here with Illinois. We cycle this system on every 80 hours. It started in 2021 and has been going since then. And we're testing this on silver carp, bighead carp, grass carp, and multiple native species. And then we're going to start working on testing this on continuous operations starting next year. We also have a study of a bioacoustic fish fence. This is at Barkley Lock and Dam on the Cumberland River in Kentucky. This is a little bit different. This is bubbles, lights, and sound. So 3 different kind of deterrent types combined into one. This cycles on every week, so on a week, off a week. And here we're testing it on silver carp, grass carp, and a few native species. This study actually concluded as it was designed in this year, but the BAF is operating continuously at this site and awaiting what its fate will be in the future. And so you just heard a lot of information about tagging and tracking fish. We do the same thing in rivers of the Mississippi River Basin. And so the way we're evaluating these systems is by looking at putting transmitters or tags in fish. We have 2 different telemetry systems, 2 different frequencies. We also translocate fish, so moving some from above a lock and dam to below to increase the probability that they'll challenge a deterrent or move upstream. And then we can use these tagged fish along with their detections on all these receivers throughout the lock and dam area, so all these yellow dots, to determine fish deterrence, fish passage, and more fine-scale fish behavior. And so for our Loch 19 study, we've tagged 200 fish there in 2021 and 2022. Additional data are being analyzed now for 23 and 24. But from these 2 years of data, we've had really limited information. So we have not had many fish come back at all. For fish that made full upstream passage, so going across or through the deterrent into the lock chamber and all the way upstream into the next pool, we had 1 pass when it was off and we had 2 pass when it was on. So limited sample size. Approaches into this area, so moving from downstream in the lock approach channel into this area, we did have 39 individuals approaching when it was off and 43 when it was on. The number of times they did this did differ, more times when it was off than on, but overall not a huge difference here for grass carp deterrence. We do see a much larger deterrence rate for silver carp in this area. We then started to look at fine scale behavior. Even though we're not seeing large numbers of fish coming back for passage, are we seeing differences in where they are within the lock approach channel? And we've just started these analyses, so it looks like there may be some differences with fish hanging out behind or downstream of the deterrent. But currently we're not seeing differences in where they're turning around. So going back downstream once they come up into the approach channel, even though there looks like there's a little bit of a difference here in more turning around when it's off closer to the deterrent versus when it's on. But we're exploring this one more. For Barkley Lock and Dam, the passage results for this, there's 75 tagged grass carp there in the 2021-2022 time period. Those that have made full upstream passage from downstream all the way through the lock into Lake Barkley, 13 when it was off, 18 when it was on. And so deterrent crossings, how many crossed this bubble sound light curtain? And that was 34 individuals when it was off and 23 when it was on. So we're really not seeing You know, a huge difference here as well. There is a difference between the number of times that fish crossed. So events like you saw for Loch 19, about double the number. This just shows when they come into the lock approach channel, when they're passing, when they're trying to make passage. And most of this passage actually occurs in this May-June timeframe. And so this is when the deterrent would need to be most effective. The fine scale results from Barclay. So we used a subset of the data from the full passage that I just showed you and looked into it further to see what kind of more fine scale behaviors are being exhibited. And so during a deterrent operation of off, full crossings of just that, the deterrent part, not moving all the way upstream, we did have 118 fish when it was off and then 50 crossing when it was on. So a little more than half. And the number of fish was 32 and 19 when it was on. It's a little less. The number of rejections, the number that tried to pass was 40 fish and 33 fish when it was on. So a little less, but a lot fewer pass attempts. So a lot fewer fish coming into that area and being rejected. All right, so again, with this subset of data, we're looking at fish within this 15 meters of the deterrent. So these two lines kind of blanket this area and seeing, you know, describing these two main pass attempts, the full crossing and the kind of these rejections. All of this is within this 30 meters, this fine scale movement. For the fine scale behavior, we found when the deterrent is off, grass carp actually enter. So where are they entering? What are they doing? They actually enter, maybe this will work. There we go. In any zone, so all the way across that approach channel, across the deterrent. Oops. But full crossings actually occur more often in the middle. But the rejections tend to enter and exit then on both sides when this is off. Oops, I'm sorry, did the wrong one here. So they enter in any zone when it's off, so you can see this large arrow, and then they exit over here on this land wall side. For on deterrents, when the deterrent is on, grass carp full crossings occurred more often in the middle. So you can see here, and then rejections were entering and exiting on both ends. So a little bit of difference in that fine scale. Overall, we're seeing the probability of a rejection increase, so they're deterred when the deterrent is on, when vessels are present in the area during the winter months, but there was no correlation with water elevation. So it's just a brief summary of the two large deterrents that we have been working on. Hopefully we'll have more data and more fish coming into the areas for both of these projects as we get to 2023 and 2024 data. And this is just some pictures of all the work our crews have done on these projects. If you have questions after this, you can contact myself or Andrea. And I think we're turning it over to Ryan.
Thanks, Mary Beth. All right. There we go. Okay, so I realize we're running late on time, so I will very briefly go through these 5 slides. So this is a discussion of a deterrent that's at the other end of the technology readiness level scale, and that we're just working on prototypes here in the laboratory, similar to where Mary Beth's work started. There we go. So motivation for this is looking at bubble screens and use of bubble screens for invasive carp deterrence. And so these bubble screens have been used to guide native species as well as reduce passage of common and big-headed carps at pretty high efficacy, 70 to 80%, but we have not, they have not been evaluated thoroughly for grass carp. So that's one of the motivations for this work, is that bubble screens can keep fish from accessing spawning areas, for instance. Typically, for population control, adults are often targeted for collection. However, you know, it's been shown through some of the literature and population modeling that targeting multiple life stages may be much more effective. So, can we also target younger life stages? And as you know, I look a lot at eggs and larvae, as I previously showed. And so one of the things that we came across is that bubble screens are also effective river filters, and they've been able to show over in Europe that they can be used for micro and macro plastics, and capturing them in basically diverting those plastics to a passive collection bin on the stream side. So we thought, can we use this for this purpose as well and have a dual purpose barrier? So that dual purpose barrier essentially would keep adult fish from being able to move up and access their spawning areas, those known spawning areas that we've been able to identify. And for the fish that do make it through, because no barrier is 100% effective, if we can capture eggs and larvae moving downstream at the same time, we'd be able to target those multiple life stages. So we set a target efficacy of about 50% for each direction. And as was mentioned, there's a seasonal deterrent that's been proposed on the Sandusky River. So we used the condition, or the characteristics of that cross section and that part of the river to guide our research. So our project workflow, we basically work through characterizing the bubble screen in a cross flow, which is a difficult environment. You know, the previous talk by Aaron talked about deterrents near lock chambers. We're talking about deterrents in open water conditions here, in open river conditions during higher flows. We use surrogate eggs to develop the initial system, basically precision particles, and we were able to get high capture rate, meet and exceed our capture efficiency that we set. We tested those with live eggs, grass carp eggs and larvae in the laboratory, and had poor results with that same system. So we had to go back to the drawing board, basically redesign the system, generate new surrogates. So we developed a new method to preserve grass carp eggs, and then rehydrate them when needed. And we also dyed them so they're visible for optical tracking techniques. And essentially, it allowed us to work year round with these grass carp eggs and their physical properties, which are very, we have to tune to the system itself. And so we ended up with a bubble screen that's oriented parallel to the flow as the best capture mechanism, and take advantage of these helical circulations that can bring the eggs to passive capture devices. And so we went back to live egg and larval experiments this last summer, and were able to exceed our 50% threshold, and even up to 75 centimeters per second of flow velocity. However, that parallel bubble screen obviously is not going to impede upstream migrating fish. And so we're separating those experiments out into separate experiments in 2025. And so we're going to proceed on different tracks with laboratory experiments for upstream passage with an oblique bubble screen, and then our dispersal and capture mechanisms for eggs and larvae on another track. And so we'll be moving towards near field scale experiments with the dispersal variant in 2025. And that'll do it for us. Apologize for going over time.
Thank you so much to the USGS team. That was awesome. A lot of incredible work. We will now go to Ryan Young with U.S. Fish and Wildlife Service.
All right, hello, can you all hear me and see my screen?
Yes, we have both, Ryan. Thank you.
All right, my name is Ryan Young, and I'm a fish biologist at the U.S. Fish and Wildlife Service Alpena Fish and Wildlife Conservation Office at the Detroit River Substation. I'm going to talk about grass carp today, specifically our ongoing removal efforts occurring in the Great Lakes. So as a little introduction to the problem, grass carp were first introduced to the U.S. in 1963 and have since been transported, escaped, and invaded natural waterways such as the Mississippi River Basin and the Great Lakes waterways as well. So these fish, when they're stocked, there's several states surrounding the Great Lakes that allow the stocking of triploid fish only, which are sterile fish. And then there's states that prohibit stocking of grass carp in general, including Canadian waters. So grass carp escape from ponds or during unauthorized releases, such as bait buckets or religious ceremonies. And when they get into the environment, they can cause a lot of damage. So they consume 20 to 100% of their body weight in vegetation daily. They can grow up to 80 pounds. And they really have some adverse impacts on vegetated habitats, fish communities, and fisheries of Lake Erie and other connected Great Lakes. The picture on the right shows one of the fish we captured this year. As you can see, it is just full of eggs. And you can see the amount of vegetation this fish has consumed. So again, they eat a lot of vegetation and they're highly fecund, which present a risk to the Great Lakes. So I'm going to start this presentation off with a definition of what adaptive management is, because that's the theme of our response efforts. Adaptive management is a structured process for making decisions and improving management practices over time. So adaptive management is useful when there's uncertainty about the best strategy for managing natural resources, and in our case, grass carp in the Great Lakes. So why is adaptive management important? These are the steps of adaptive management process. We assess the problem, we design a strategy, we implement that strategy, we conduct monitoring, and then we evaluate our efforts. And essentially, the monitoring and evaluation steps provide feedback on the success of our management actions. And then we adjust our strategy and then see where we're at. And in our case, it's a 5-year management strategy, which I'll get into here shortly. So the Lake Erie Grass Carp Adaptive Response Strategy was developed in December 2019. It's a 5-year strategy from 2019 to 2023. And key management considerations of this initial plan were likely sources of natural reproduction and pathways of additional introductions of grass carp, the accuracy of grass carp abundance estimates in Lake Erie, the efficiency and effectiveness of agency grass carp removal efforts, with specific objectives. Really the first one, to improve the collective understanding of grass carp population dynamics, behavior, and impacts in Lake Erie to inform effective management actions. Second objective was to implement controls to minimize population expansion by removing fish and or blocking access to preferred habitats. And the third objective is to minimize the likelihood of introduction and establishment of new breeding populations of grass carp in the tributaries and nearshore areas of Lake Erie and Lake St. Clair. This strategy was implemented during 2019 to 2023 by federal and state agencies, working in partnership via the Great Lakes Fishery Commission's Lake Erie Committee. And future revisions to the strategy will be informed by changes in grass carp population status in the Lake Erie Basin, in the sources and pathways of new introductions, and in the availability of new science, tools, and resources for detection, monitoring, and control. So this was the first iteration of the plan. The next slide shows the second iteration of the plan, but really focuses on the differences between the first and second. So with key management considerations, you see the efficiency, effectiveness of agency grass carp removal efforts, but it includes the societal acceptance of those removal efforts. How are we perceived in the eye of the public when we're conducting electrofishing operations daily, or we're pulling nets that have large amounts of fish or result in relatively high bycatch. And then the second key management consideration that is different than the first plan was really to address the uncertainty of the density thresholds at which grass carp herbivory becomes detrimental. The objectives remain the same as they did in the first plan, but with regards to implementation, knowledge gained during implementation of the initial strategy guided development of the second strategy with technical review and advice provided by the Grass Carp Advisory Committee that was formed by the Council of Lake Committees in 2020 and populated with the representatives from partner agencies. So again, future revisions will be informed by changes in the status and trends of extant grass carp in the basin and the sources and pathways for new introductions and evidence of adverse impacts from grass carp herbivory on aquatic vegetation and the availability of new science, tools, and resources for detection, monitoring, and control, and from societal responses to management efforts. So the Great Lakes Grass Carp Advisory Committee is really, it oversees our working group, and I'll show a schematic that was shared earlier in one of the other presentations. But the task groups are charged with undertaking and coordinating special focused activities needed to achieve GCAC and individual lake committees' objectives. So we have a data management task group, a fieldwork task group, telemetry task group, modeling task group, barriers and deterrence task group, and early life history task group. And you've seen some of the research coming from those various task groups in the earlier presentations. So relating to our organizational chart, this was also shown, but in a different schematic. I'm really going to focus this presentation on the removal aspect of this. Essentially, all of the field data collected by removal teams informs research, and then the research informs removal efforts and helps us target new areas and become more efficient in the work that we do. So I first want to acknowledge the Michigan Department of Natural Resources, Ohio Department of Natural Resources as the state jurisdictional authorities, University at Buffalo, and the University of Toledo as well, for all their hard work they do with their boat crews and strike teams that are conducting work all around the Great Lakes. So focusing on the key management consideration of efficiency, effectiveness, and societal acceptance of agency grass carp removal efforts, our intent is to prevent grass carp from attaining densities that cause adverse impacts. So I'm really going to go into an overview of the primary removal methods, and then I'm going to dive into each one of those with specifics. So boat electrofishing, trammel nets, hoop nets, and gill nets. This picture on the right here shows a large-scale planned action event in which multi-agency boat crews get together during a high-flow event, and they use boat electrofishing in tandem to try to capture as many of these fish as possible as they aggregate for spawning events. Boat electrofishing, our fleet of vessels are aluminum. They have unpainted hulls. They're typically two boom electrofishers that are 18 to 22 feet in length, and they're outfitted with two primary types of pulsators. So the Smith-Root Apex box and the Midwest Lake Infinity HC 80 amp box. There's some slight nuances between those two different pieces of equipment, but essentially you can input the same waveforms and voltage into these boxes, and they produce very similar results. The only benefit of having the Apex box is you could stick a USB drive in the box and download the data after a transect. It also records the GPS boat track coordinates of vessels during a transect as well. Most of these vessels have Honda 7000 inverter style generators. They're quiet, makes it easier for operators and detonators to communicate. during transects, and there are typically 1 to 2 dip netters on board, depending on availability of crews. So for the first case study, it was a laboratory study using juvenile grass carp to determine electrofishing efficiency. Essentially, this group of researchers from the Fish and Wildlife Service conducted this study in a lab, and their results indicate that using a pulse direct current waveform at 60 hertz and 24% duty cycle and 80 hertz and 30% duty cycle may be the most effective. This really drove the initial response efforts for our team, in which case during these large scale planned action events that I mentioned earlier, we would use that 60 hertz, 24% duty cycle setting with the pulse direct current waveform. And then we would use a electrofishing goal table that was really meant for several cool water and warm water fish species. So it isn't species specific and was recommended by Miranda 2009. And so we noticed that use of this electrofishing goal table resulted in a lot of fish escaping, and it kind of opened up some questions for us on, are these settings effective for grass carp as we thought they might be? The next study I'm going to highlight really was a field study that used occupancy modeling and surrogate taxa, such as common carp and buffalo species, smallmouth buffalo and bigmouth buffalo, to compare the effectiveness of boat electrofishing and the combination method of boat electrofishing within and around a trammel net enclosure. And so results from this study indicate that detection probabilities were higher for the combination method than for boat electrofishing alone. So the purpose of presenting these case studies is to show an evolution of our methodology for response crews to capture these fish. So started with electrofishing during these planned action events, and then we're starting to take a deeper look into the gear types of electrofishing and trammel nets to see if we're using the most effective methods out there. So this study was published in 2022. And then the third case study, it's not really a study, it's more opportunistic research that used adult grass carp to determine threshold settings in the Cuyahoga River, Ohio. And by threshold settings, I'm talking about the amount of electrical stimulus applied to water to cause a given response, such as taxis or immobilization, where a dip netter on the front of the boat could net fish and capture it. Really, we didn't expect to come across a large number of fish in the Cuyahoga River in 2019. It was a combination of luck, but during our first few days out there, we noticed a relatively high number of grass carp, and we had relatively clear water, which allowed us to take some notes on and observations on fish response. Previously, we weren't able to do this in the Sandusky and Maumee Rivers due to the high turbidity. And so ultimately, results from this opportunistic research were used to generate a refined electrofishing goal table with voltage, current, and power goals specifically for adult grass carp. And this is these settings are what we have used since 2020 across the Great Lakes to capture fish. The trammel nets with specifics, there's two types that we've used over the years. These are 600 feet long by 14 feet deep, or 600 feet by 8 feet deep. Trammel nets are different than gill nets in that there's two outer panels that are 16-inch square mesh, and then the inner panel is a 4-inch square mesh. And essentially, the trammel nets, as the fish hits it, it gets caught and bagged. It doesn't get caught by the gills. And so it works a little differently than a traditional gill net. Hoop nets have also been used, and these have been 3 feet in diameter with 7 fiberglass hoops. They're 12 feet long. They have 2 throats with 1.5-inch square mesh, 25-foot leads, and 10-foot wings deployed at 60-degree angles. Gill nets that we have used, I focused really on the 2 types of gill nets where we had a significant number of sampling events. So we used standard nets that were 300 feet long by 12 feet deep. These were 4-inch square mesh And these were 8-ply twisted monofilament that were 0.2 millimeters in diameter. And then the second net type I'm going to be covering in this presentation is an invasive carp net. It's 450 feet long by 12 feet deep and 4¼ inch square mesh with 0.9 millimeter monofilament. So much different net than the twisted mono net. And we've noticed a difference in the effectiveness, which I'll get into here in a few slides. So we use all these different gear types to collect data on grass carp. And there's 13 boat crews that are out there today. So all of the data that they've collect go into a database. It's a shared multi-agency database. This is the initial database structure. And really there were, in 2018, there were 2 agencies primarily conducting targeted removals. That was the U.S. Fish and Wildlife Service and the Michigan Department of Natural Resources. In 2019, the University of Toledo came online, and we had a 3rd boat crew out there. We started collecting data on paper data sheets, entering into Microsoft Excel, then developed a Microsoft Access database to accommodate the multi-agency data. And then we quickly found that, you know, over the years of 2020, we had 4 crews out there. And then 2024, fast forward, we have 13 boat crews out. So a lot of data being collected, a lot of pieces of data, and it was very messy how we how all this data fit together. So fast forward, we had to implement some database management. And we have a much more streamlined process now for collecting data on various components, whether it's ploidy, whether it's aging data or diet data, and all this data gets linked into ArcGIS Online Survey 123, goes through this process of review from folks with various data roles and responsibilities, ultimately gets QA/QC'd, and then is available to agencies to use for research. So here's the current data workflow. We have field work within 48 hours, data is submitted. Within 10 business days after that, we have QA/QC requests, and a QA/QC lead is designated by each agency. They go through and check the data. That data then gets validated by the data stewards and data custodians, and then the data becomes available. And so right now we have a clean version of the database from 2020 to 2024. The data from 2018 and 2019 was so different than what we do today that it kind of exists as in its own database, but is still available and has still been QA/QC'd. It's just in a different format. So now I'm going to get into the numbers here. So for our collective agency effort from 2018 to 2024, the red dots represent effort points throughout the Great Lakes. The darker dots represent higher numbers of transects that were deployed. And so as you can see, the majority of our effort has really been electrofishing, over 4,484 hours. We've had a lot of trammel net sets collectively, about 1,800 trammel net sets. Hoop nets, it's a little under 1,000. These invasive carp gill nets, the 450 feet by 12 feet, only 197 net sets that were deployed in 2024, and then even fewer of the 300 foot by 12 foot gill nets. I want you to pay attention to the catch here and specifically look at those invasive carp nets that are 450 feet by 12 feet. So the yellow diamonds represent capture points throughout the Great Lakes. And as you can see, the majority of fish we've captured using electrofishing. The second most effective gear type are these invasive carp nets. And we've only had 197 net sets, but we captured 177 grass carp, which is incredible. And I'll get into the catch per unit effort in a few slides here. Trammel nets, not very effective. We've caught a few fish over the past 6 years or so, but they have a lot of bycatch, and they're very, very difficult to work up, and there's a trade-off there, and the amount of time it takes to work through those bycatch, those large amounts of bycatch, than using boat electrofishing and being able to cover more ground in the river. So when we're looking at our total Great Lakes removals from 2018 to 2024, you see an increase in captures over time. So some of this is reflective of the number of boat crews that are out there. 2019, you can see a large number of fish captured, but 74 of those fish were captured during 12 days of sampling in the Cuyahoga River, which is what we used for that electrofishing threshold study. So the overall trend is an increase in capture numbers over time. You can see that there's been 2022 to 2023, there was few captures in Lake Michigan waters, but really not that many captures here in 2024. So Lake Erie seems to be the hot spot for grass carp, and that's where we continue to focus our efforts. These are the number of grass carp that have been released in US waters. So these are fish that were either captured that had an existing tag in them, or they were new fish that we were able to surgically implant acoustic tags and satellite tags into in coordination with our partners. So you can see in 2023 and 2024, we're taking advantage of these higher capture numbers to try to get as many of these tags out there to get useful information on their movements, which will inform, future removal efforts. When you look at the specific Lake Erie tags in 2024, you can just see the breakdown in the table on the top left there. In the Sandusky River and Maumee River, we got the majority of the tags out, including those satellite tags. And then the image below that shows the movements of one fish and what that satellite tag data looks like. Satellite tag tagging process, you can see the end result on the far right there. And then in the middle picture, you can see that is implantation of an acoustic telemetry tag. When you look at our grass carp CPE over the duration of 2018 to 2024, you can really see that we've been pretty consistent from 2019 to 2023. In 2019, again, you have those large number of captures from the Cuyahoga River. You remove those and you have about 0.15 fish per hour, which is the average across years there. But in 2024, you'll see that it's lower, and that likely represents a shift of our efforts from electrofishing to these to this gill netting, the invasive carp gill netting. Those gill nets had a mean CPUE of 0.9 fish per net set, which is just much higher than any of the other nets that we've tried in the past. And so it's promising that we can use telemetry data to inform our gill netting efforts. And that's what I'm going to get to in this slide. So gill nets are typically deployed at a 45-degree angle from the shoreline, stretching out towards the middle of the channel. So compared to electrofishing, you're covering a lot more ground, where electrofishing you're most effective between, up to that 9 feet depth. And beyond that, in these 12, 14-foot holes, electrofishing just really isn't very effective, and especially in areas that are wide and not confined, such as areas in Farnsworth on the Maumee River. So we've used telemetry data from our VR100 unit. So this is basically an acoustic hydrophone that we stick in the water and listen for fish before a transect. And we've detected a number of unique individuals in this Farnsworth Metro Park location. And I will mention that these points aren't exact points of where a fish was. With the hydrophone, a fish could have been within 1 kilometer distance from where we were listening. And so, but either way, it identified river kilometers that we could target to set these gill nets where there's higher detections of unique fish. So sites for these overnight gill net sets were selected using available bathymetry and telemetry data. And then we've also reached out to our USGS partners for some satellite data to see other locations where we might want to try to deploy these overnight gillnet sets. Very low effort on our part to deploy these. It takes about 5 minutes to set, and on average 30 minutes to pull the nets after a 24-hour soak period. So here's our invasive carp net summary for 2024. Couple tables here. The top left table is really focused on location. As you can see, the majority of our netting effort has occurred in the Sandusky and Maumee River, and both have resulted in relatively high CPUE than what we see with other net types. We've also had some success setting these nets with limited effort in some of the other tributaries of concern for grass carp spawning, such as the Grand River, Ohio, and Huron River, Ohio. So there's some promise there to implement that on a larger scale. The one thing I want to point out with this location figure is that this work wouldn't happen if we didn't have clear and effective lines of communications between us and partners. So within noticing how effective these nets were after our first week of using them, we immediately sent three of 10 of our nets down to the University of Toledo so that they could start fishing these nets in the Sandusky River to see if they had similar success. And what they found is that these nets were really effective in areas of the Sandusky River, such as the lower river, where we just haven't had much success at all capturing fish, electrofishing, or in any gear type really. So knowing that fish utilize the lower river and not being able to catch them with our traditional gear types, having this new innovative method where we could set nets overnight is really promising for the future. The figure below that really breaks down our net sets per month. And really what I wanted to show here is that we caught a lot of grass carp in August when traditionally electrofishing catches were very low and we just have had a hard time in general catching fish. September was also pretty successful, and even October, but you can see that our catches have declined. Some of that is probably attributed to the fewer net sets that we've had over that period. When you look at these nets, what does the bycatch look like? Well, in the Sandusky and Maumee rivers, you can see that pretty low bycatch. It's under 20 fish per net. And when you're looking at high catch per effort of grass carp and low bycatch, that's kind of the golden ticket that we're looking for. So again, trying to figure out ways to use this new gear type and become more effective. The majority of fish that we captured as bycatch species were flathead catfish, buffalo species, and common carp. But it's interesting to see that 5 to 6% of our total grass carp catch in the Sandusky and Maumee rivers were grass carp. When you look at the ploidy, or essentially the fertility of these fish, you see that the majority of fish captured over time, over that 2018 and 2024 period, we're leaning towards a more diploid dominated system. So there's still a large number of triploid fish in there, but we have been really effective at reducing small aggregations of triploid fish, and the Cuyahoga River is a really good example of that. Out of the 74 of those fish, 64 of them were triploid fish, and we have gone back in consecutive years and have seen a continual decline of fish that we detect in that system significantly. I believe we had 8 fish in 2020, down from 74, 5 fish in 2021, and then even fewer fish in 2022. So pretty effective at removing pockets of triploid fish, but triploid fish are still a problem. They still live a very long time and they still eat a lot of vegetation. So it's critical that we, remove all adult fish. When you look at the otolith microchemistry results from 2019 to 2022, This was a cooperative agreement that Fish and Wildlife Service set up with the Southern Illinois University and a graduate student there. We sent them a large number of fish, I believe 498 fish. A lot of those were triploid fish, but 219 of those were diploid fish. And so we wanted to figure out where those fish were coming from. Unfortunately, I cannot tease apart the Lake Erie diploid fish from the unknown ploidy fish. So we were only able to really focus on what we knew were diploids, and those unknown ploidy fish kind of didn't allow us to tease apart which were triploid versus diploid. But a little introduction to strontium. Strontium is a trace element that is readily substitutes for calcium in the otolith's crystalline structure and allows researchers to analyze the varying concentrations of strontium within the otolith to infer information about a fish's environmental history. And what we found is from these 219 fish, clusters and broad ranges of strontium calcium values among samples suggest multiple sources of introduced escape fish in the Lake Erie Basin, and some of these are capable of long distance dispersal. Where does this long distance dispersal example come into play here? And I want to point out the St. Joseph River, where one wild fish was captured, and the strontium calcium isotope data suggested that that fish likely originated from the Lake Erie Basin. So that may be an example of a fish making a long trek all the way from Lake Erie to southern Lake Michigan in the St. Joseph River. And what was interesting about the data for that particular fish is that fish made the journey in the latter part of its life. So these fish are long-lived. They can live up to 30 years in the Great Lakes. And what we see with the telemetry data is, the tags are only active for a certain amount of time. And I wonder, as those tags expire, if we see that distance that fish typically move increase throughout their age. So this suggests that may be happening. The second big result from this project was that we found that 97 fish likely originated from the Sandusky, Raisin River in Michigan or Portage Rivers. 87 fish came from the Lake Erie Western Basin, but we weren't able to tease apart which rivers. 23 fish could have come from any of the basins, either the Western, Central, or Eastern Basin. 7 of the wild fish came from the Sandusky River. And what's new about the findings from this study compared to previous studies is that a higher number of grass carp were found to originate from the Central or Eastern Basin east of the Huron River. I believe previously 1 fish was captured which fit that category. So again, this type of work is really useful for us to determine natal origin of fish, and also highlights that some of the diploid fish in Lake Michigan, did come from aquaculture. So you still have aquaculture diploids making their way into the system. So for my conclusions, catch is increasing because we are using innovative gear types and methods, such as invasive carp nets, and are exploiting new areas previously inaccessible by our traditional electrofishing boats. So this is using small john boats or rafts to fish gill nets. It's not as easy to do, but we're able to access new areas such as the image on the right. This is Van Tassel Wildlife Area. It's upstream of Farnsworth Metro Park area. The pink triangles represent gill nets that were set with no captures, and the various size circles there represent up to 8 fish captured in a net. I believe 3, 5, and 8 for the various sizes. So curtailing natural recruitment, further introductions, and spread of grass carp is necessary for successful population control. For future work, given that these invasive carp nets have a lot of promise, we want to conduct a gear efficiency comparison study with partners at USGS. They're leading that study, but we're specifically interested in the 450 feet by 12 foot nets and a number of other passive gear types. our office in Detroit will be conducting a fleet standardization study. So essentially, we're going to look at existing data for electrofishing vessels, for each electrofishing vessel within each agency strike team. And we want to see whether or not the threshold data for grass carp captured on those individual vessels is similar. And we want to see if there's differences between those vessels, or if they're all fishing around the same effectiveness, which is ultimately what we want, because we want crews to be following the same protocols, which we have one protocol. It's a unified protocol that's shared between all strike teams, and it's just a way to evaluate how folks are doing after the fact and make adjustments as necessary. The third study that we're going to be participating in 2025 is an electrical field mapping study. But we have a few vessels in our Detroit office that we're interested in doing this. So essentially, when you have your two booms deployed, what does that electric field look like under a variety of conditions and static conditions when you're fishing the lake? Does that look different than when you're working in a high flow event and the cathode droppers on the front of your boat are drifting back in the current? Does that electric field change, and could that potentially explain why we see differences in boat electrofishing catch during high flow events versus during non-flow events? We're going to look at traditional electrofishing boats, but we're also going to be doing this for our raft electrofishers, which gets me into our raft electrofishing. We've deployed two types of rafts. One is a non-motorized raft. That's a Smith-Root ECAT raft on the right. We've outfitted that raft with a Smith-Root Apex box and the Honda generator. It's two boom electrofisher. We've captured a few grass carp in this during some drift surveys last year in 2023. We spent the majority of this year trying to change the cathode design on that vessel to get more power going to the anodes so that dip netters on the front of the boat can net fish. And then that information has been very useful for informing the development of a University of Toledo motorized raft on the left. You can see that they have an interesting cathode design there, washing machine cables that are stainless steel hanging over the sides on either side. And then we've had to adjust this design to increase the power going to their anode, and preliminary results suggest that our modifications have been effective. And so we're going to use that information when designing our own motorized raft for Fish and Wildlife, which is also this Zodiac style design here, except for it's going to have a aluminum frame that you can take in and put out of it. So that's what we have in the works for 2025. And then I want to acknowledge everybody from our partner agencies, past and present. This has been a monumental task to try to manage highly elusive invasive species in a large system such as the Great Lakes. And it takes a lot of people and a lot of brilliant minds to get together to try to effectively manage the species and prevent them from becoming established. So thanks again to everybody who's involved in this work. And that is all I have for my presentation. Thank you. If you have any questions, please reach out to me via email.
Thank you so much, Ryan. I didn't even have to give you a warning. You finished right on time. All right. So I'm just going to open it up for questions now. I don't see any questions currently into the chat. But we'll just, give it a few minutes. If anyone has any questions, just type it into the question box. And then Ryan, if you want to stop sharing your screen, you can. Okay, it doesn't look like any questions are coming in, so I guess that is going to do it for today. Thank you all so much for attending, and we will have more of these webinars in the future. Next year, we will be having some webinars on harmful algal blooms and water chestnut management. And as I had mentioned before, I see that the recording will be posted on the NEAPMS website at www.neapms.org. So again, thank you all so much. And thank you to the speakers. They were some really great talks, and I look forward to sharing this recording with a lot of people. And that's it. Thank you so much.
Thank you, Lindsay. It was great to participate.